Center for Biotechnology Research.
Well, I am the director of the Interdisciplinary Center for Biotechnology.
Okay.
However, I'm from the University of Florida, and so ICBR is the biotechnology center that does all the sequencing and proteomics and different kinds of things like that for the University of Florida, and so I have the privilege of directing that group, that facility. But I'm a professor at the University of Florida, and so my primary role is as a researcher that does plant research along with my colleague Rob Furl.
So then, Professor Pohl, I have some questions for you.
Happy to.
You said you worked in Antarctica, right?
Yes.
Can you just give our viewers a bit of information about it?
So two things. Working in Antarctica is an extraordinary experience. It is one of those things that we scientists do to help practice what it's like to be in an extraordinarily isolated environment, much like practice being on the surface of Mars. And the other thing that allows you to do is it can give you this sense of, when you look out onto the ice of Antarctica, you walk off the plane for the first time, and you have this feeling of humans don't belong here. Humans are guests here. And the kind of awe and wonder that you get by first walking off the plane in Antarctica is one that I hope that the astronauts get when they first walk off onto the surface of Mars, right? But how you get there is pretty much how you get any kind of research going, is that for us we're interested in doing what will it take to take plants off planet. And so we had collaborators with the German Space Agency, and we worked in the Future Exploration Greenhouse, the FEG, at Neumayer Station. And we were part of the team to help, what would it take to set up a type of greenhouse in an isolated environment that could collect data from how plants are going that wouldn't include an astronaut per se. And so what Rob and I were working on was a special type of imaging that allows you to take special images of the plants that can report back to you on their state and their physiology. And one of the things that we found while we were down there is that by using these specialized cameras, we found that one of the trays of plants had some imbalance in the nutrient solution, and we could see that from the images before you could actually see it in the plants that they were wilting or something. And so that kind of technology is what we were doing down there.
So practically preparing for Mars.
Yes.
Mentally.
That's right. And practically in some ways too, because you have to be innovative when you don't have all the resources that you can just go down the road to the store to pick up supplies and things. And so it's also practicing and challenging of how to think in those kinds of isolated terms.
In your presentation a few hours ago, you spoke about the gene expressions.
Yes.
And there I have some questions for you too. Or because you told us that they have like a toolbox of genes.
Right.
And how could they know, or how could they be prepared in their genes? How could they be prepared for space? How do they have the best genes?
So that's a very good question, and the answer is they can't. They can't be prepared for space because they didn't evolve with space. They don't have that in their lineage. But what they can do is they can say, and I'm being very anthropomorphic here, right? But a plant responds by saying that, all right, I'm detecting some difference in my environment. What do I have in my toolbox that I might be able to, that seems familiar to deal with? And so, for instance, a lot of the genes that are differentially expressed in space are associated in normal cases with pathogen infection or with stress responses that are associated with fungi and things. And you ask, well, is it because they're being attacked by bacteria or fungi? And the answer is no. But one of the things that happens when you are infected by a fungus is it pokes a hole in your cell wall. And so when the cells and so our hypothesis is, in microgravity, the cell walls seem to relax a little bit, and that relaxation elicits the same kind of sensation of, oh, I think I'm getting attacked by a pathogen. I'll turn on these genes. And so some of the genes that we think that some of the genes that are induced in spaceflight aren't really necessary for the survival of plants in space, but they're a response to things that are somewhat familiar in other aspects, and it constitutes a, quote, spaceflight response. Now, one of the things that we have learned over the sort of the decades of not just Rob Furl and I doing these experiments, but our whole community, is that the better gardeners we become, in that we use the hardware to help mix the gases better, to help control the water tension and stuff in the growth medium, the better we are at making these kind of habitats, the better plant to do, and the fewer and fewer genes they turn on in response to spaceflight, especially plants that are older as they've kind of adjusted over time. We also see a big difference between seedlings that are only 4 days old. There's so much data I didn't show, but just a 4-day-old seedlings versus 8-day-old seedlings versus 12-day-old seedlings. The longer you are growing in that habitat, the more sort of relaxed you get, and like, okay, those genes didn't seem to do any good for me, so I'll stop wasting my energy of turning those things on, and I'll start settling down. And so when we grew plants for long periods of time in the advanced plant habitat, and so we had those plants that we used for the epigenetic studies and things, we were quite surprised that a lot of the genes that we see are being differentially expressed in those sort of mature plants that are well adjusted now to their spaceflight habitat. There's not as many genes as we see in the small plates, in the Petri plates, where they're only growing for short periods of times, because they're starting to understand, if you will, again being anthropomorphic, that turning on all these genes for cell wall metabolism, they don't really, quote, need it, and so they stop investing the energy to turn on those kinds of genes. At least that's how we think about it.
To your other experiment with the moon dust and the moon soil.
Yes.
How was the feeling to plant something like this?
It's amazing. And even when I've talked about this in very different kinds of ways now many times, but even standing up here just a half an hour ago, I could feel it in my chest, right? I could feel that emotion, because it takes Arabidopsis a couple of days to germinate. And so Rob and I did all those plantings, and because we had to kind of keep it secret from, NASA made us keep it secret until we actually published it. And so we couldn't bring in the normal number of students and postdocs and things to do this work. And so Rob and I did a lot of that work ourselves. I mean, the putting it all together and stuff. We had some help, but it was, but we were very invested in it from just personally. And so we got all those things planted, and we put them in the growth room, and we had a special lock room and everything that we had to grow them in. And it's like, okay, they're not going to germinate for at least 3 days, so we should just chill and not go in there. But of course we couldn't do that. And so on day 2, we went in there, and they were already starting to germinate on day 2. And when we walked in there and saw just a sea of these little tiny green seedlings, it really just brought tears to our eyes, because you think about this is the first time that terrestrial life has grown in an extraterrestrial material, and we're seeing it. We are the first humans to see this, and it was really emotional, and it was also emotional thinking that kind of part of, small part, but of history, right? That this is, anytime you have the privilege to do something like that, it's a real honor to be part of that history, and it sort of struck us all at once at that moment in a very strong way.
So you took the moon dust and just mix it with water before planting the seeds, or?
Not quite. So it didn't have time to really go into all the details, but we had practiced, like I said, for essentially 10 years on, because the first time we wrote the proposal, NASA said, no, we're not giving it to biologists. You'll make it all messy and stuff. Just practice with simulants. I'm sure you'll get the same information. And so we practiced with simulants, and we bottom line is we got very good at using the simulants to create a growth habitat with a very tiny amount of material. And so we had in those trays, we had a layer of what's called rock wool, which is essentially a glass sponge that will wick water very well but doesn't retain water, so it won't make it muddy. And then we had a very thin filter that allowed so the dust wouldn't go through the filter, and then 900 milligrams of the moon dust. And initially, what always worked for the simulant is you just put that tray in a tray, and we didn't use plain water, we used a very dilute nutrient solution so they'd have something to work on, and then we just wick right up. But the lunar dust didn't do that, and so we were confounded with, well, now what are we going to do, because it doesn't work like we had practiced it. And so what we wound up doing is we had to stir it first inside those little wells with a little plastic Petri pipette tip in order to get all the surface area wetted for the first time, and then it would draw up and we could water the plants over time that way. So we didn't use just plain water, we just used this very dilute nutrient solution, but we used that same dilute nutrient solution for the controls, which was the JSC1A regolith simulant, as well as the 3 Apollo sites.
Wait, but the start base that died in the end of 2 weeks, right?
We let them grow for 3 weeks, and then we harvested for 20 days actually. So maybe I misunderstood your question.
Oh, because you showed us, I think, 4 of these plants.
Yes.
The 3 Apollo missions and a base one.
Yes.
And the base one was the down part.
Right, the ones that were all beautiful and green. Yes.
But there was one that after these 20 days.
Yes. Some of them didn't grow. Now I can say. So we had 3 different sites. There was Apollo 11 site where the first astronauts landed, Apollo 12, and then Apollo 17. The plants that most of the plants that grew on the Apollo 11 site were those little tiny ones that didn't grow well. The Apollo 12 they grew very well, and Apollo 17 was kind of mixed. And so when we looked at the patterns of gene expression on those plants, we could see a lot of different kinds of genes that were used by the ones that were very stressed, like the Apollo 11 ones versus the Apollo 12 ones, where they grow really pretty well. But one of the things I didn't talk about, which I was hoping somebody would ask me because I just forgot, is that what's the difference between Apollo 11 and Apollo 12? What's the difference between the sites? Why would the regolith be so different? And the reason is that the Apollo 11 site is 1 billion years, almost 1 billion years older. It's 900 and some odd million years older on the surface of the moon than the Apollo 11 site. And so the regolith that the astronauts from the Apollo 11 mission collected has been exposed to the solar wind, micrometeorites, everything. And so it's much sharper, it's much more reactive. It has a lot more of this material called nanophase iron that's very reactive and plants hate. So it was far less compatible to plant growth than the Apollo 12, which was much younger and less. But even with the very best, healthiest plants in growing in the regolith, like in the Apollo 12 ones, there were still a lot of genes that were differentially expressed, that those plants were still struggling to grow in this kind of new environment, even when they looked pretty okay.
Thank you for the interview. Thank you for
you're
your
welcome.
valuable time.
It was absolutely my pleasure. Thank you very much for your interest.